That's a man who shows his commitment to his woman. You know who that is? You know who that is? That's Estes Kefauver. And that's Estes Kefauver. Let's see here. When the date was this photo? 1956. Estes Kefauver, senator from the state of Tennessee. He became friends with Al Gore's dad, Al Gore Sr., actually, who was also a senator from the state of Tennessee. Kefauver ran many of the hearings in the United States Senate regarding how the government was going to be successful or how to be successful in cracking down on organized crime. He also proposed some legislation in the late 1950s regarding drug pricing and accountability for drug and pharmaceutical manufacturers. I guess they're the same thing. I don't know. In the United States of America. And those ideas were met with resistance by many other people in the United States Congress, as you can imagine, because especially Republicans at the time, they were like, you got to stop this, Kefauver. Now, Kefauver was actually a very beloved person in the United States. It's amazing how almost nobody in modern media, really think about it, almost nobody in modern media, and especially almost nobody in modern citizen journalism, nobody, will mention a man whose work resulted in the FDA being amended in 1962, 1963, in such a way that drugs had to be not only safe, but safe and effective. The whole concept of efficacy came out of those 1962, 1963 Kefauver-Harris FDA amendments. It was this thalidomide story, this drug which was being reused by women who were struggling with pregnancy. Many women were encouraged to deal with the discomfort of it, to take different types of pharmaceuticals which were repurposed from World War II Nazi Germany. One of these, of course, was thalidomide, and it was observed that there were developmental impacts on the fetus during pregnancy when thalidomide was used during certain periods of time of pregnancy, such as missing limbs, for example, quite severe. And the observation that there were children being born with these particular defects, as those defects were happening, those birth defects were happening, and they were happening mostly in Europe and a little bit in Canada, and they were being observed really before that started to happen in the United States. Now, the FDA, I think in the United States, although there were some people born in the United States impacted by thalidomide, the number was, I believe, very small. And it's because the FDA actually acted. I saw somewhere on social media recently someone speaking very negatively about the FDA, and the proof was the thalidomide story and all the thalidomide babies. But most of those actually happened outside of the United States. And the FDA kind of worked. Now, it was in the process of reassessing what it needed to do and adding efficacy, and the review of the Kefauver-Harris amendments. And by the way, by the time those amendments were done, it was not even close to what Estes Kefauver actually had hoped for. But, you know, that's how the sausage is made, right? You take whatever you can get. But he was still happy. President John F. Kennedy was happy. It's noteworthy that when it was first discovered that this thalidomide was being correlated with birth defects in Europe, the United States Department of Justice, which was run by Robert F. Kennedy, dad of current HHS Secretary Robert F. Kennedy Jr., they were curious how to assess this, and of course they're also assessing these amendments for the FDA regarding safety and efficacy, which are being observed in 1961, 1962. So the United States Department of Justice Antitrust Division hired its very first patent attorney, a guy named Les Weinstein, father of Brett Weinstein, you know, Dark Horse podcast. And you can actually still find on Ancestry.com, and I downloaded and saved a copy of it. You can view it on his the Wiki type page I have for him on Houstonic.live or HoustonicITS.com. His ticket where he went to the Netherlands, it was in 1961, at the same time worldwide news was flooded with information about this tragedy of thalidomide in Europe. So Brett Weinstein's dad was integral to this. Now, I have absolutely no evidence at all, and I've never made a suggestion that his role in that was at anything nefarious. But I suspect that the experience of Les Weinstein, Brett Weinstein's father, of working in the middle of this thalidomide scandal, and it's a worldwide scandal, at the same time the FDA is being reassessed and amended, and thalidomide is being reviewed within the United States. I think he had exposure to a lot of the inner workings of antitrust and the FDA and the role of the Commerce Department, and I think it was a very revealing experience for him. And I don't blame him for realizing, wow, this is an amazing place to be as a patent attorney. If I was Brett Weinstein's dad, I would have done exactly the same thing. Like, this is the place to be. This is a future which is going to be, it's going to be endless work. It's going to be job security. I might be able to make a difference, with these particular decisions. And he encouraged his children to take particular routes as well to help with their own visibility. So I'm simply highlighting. It might have been somewhat chance that Brett Weinstein's dad wound up being that first patent attorney. I suspect there really wasn't an abundance of patent attorneys back in the early 1960s. It was still a little bit evolving. I don't see the family connections there. Sometimes you're in the right place at the right time. So there's no crime there. There's nothing nefarious. But I am highlighting that Brett Weinstein damn well should have known a whole lot more about the FDA than he claims to have, and about the nature of intellectual property, and probably even the nature of coronavirus himself. He should have known. He did know. There's no way he could not have known. So he was definitely playing along. Anyways, I just felt like showing this photograph here. So the last thing I'll add, and I mentioned this before, and this is one thing. It's amazing that more independent citizen journalists don't mention this. Everyone loves to talk JFK assassination. It's a whole field. It's a subject. It's a cult almost. And Estes Kefauver, someone who was very important, the stuff he was working on, and his relationship with JFK, Estes Kefauver, I believe, was poisoned and basically killed on the floor of the United States Senate only 12 weeks before JFK was assassinated. Has any other senator died in Congress? I don't think so. And it would be one thing, if he felt a little woozy, maybe went to the hospital, wound up having a heart attack. But no, he dropped and was just about dead. They took him to the hospital. They revived him a little bit, and then he died 24 hours later. They said he was a smoker. Lung cancer doesn't cause you to just die. Okay? Yeah, I guess maybe he could have had a stress heart attack, but it seems unlikely. And matter of fact, he looked relatively healthy for a 59-year-old male at the time. 58, 59, whatever he was. It doesn't make a whole lot of sense. It's most likely that the man was poisoned right on the floor of the Senate. Every JFK assassination conversation should include Kefauver. As a matter of fact, Kefauver might have actually been the public favorite to become the next vice president upon JFK's death. Because it was rare that after JFK was assassinated, Lyndon Johnson became president, the vice president became president, and then they left the vice presidential seat vacant. You know why? Because this man was so popular in the United States of America, they would have wanted him to become the next vice president. Kefauver was winning primaries to become president of the United States of America. Now, he might have been a bad president. I mean, there's some suggestions there that perhaps he was maybe more of a collectivist than some people would like. I haven't read volumes and volumes of books about Kefauver, okay? What I do know is that the everyday blue-collar American from many states really liked this guy. He wasn't an elitist. He didn't come from big money. He remembered names. He was very dignified. He didn't speak dirty. And obviously, he loved his wife. But I think a lot more could be learned by simply keeping Kefauver as part of every JFK assassination assessment. And it never is. Probably because it's much sexier to think of the JFK assassination in terms of, nukes and commies and sex compromise, rather than just how incredibly important pharmaceuticals and genetic research was. No one wants you to think about that. So, I'm calling this session, of course, they censored Haseltine. The September 7th or September 9th book, Germs, as we're covering in this 280 series, okay, a significant portion of it, 30%, maybe even 35% of the book has been omitted from the Amazon Audible audiobook. All right, you've heard me say that at least 100 times so far if you listen to this series. And one name that came up in a previous chapter, that of course was the entire chapter, chapter 12, which I think is called The Future. The entire chapter has been omitted. The entire chapter has been omitted. In that chapter, we bump into this name called Haseltine. William A. Haseltine. Actually, it was an earlier chapter. It was still censored nonetheless, okay? 232 to 233. So I'll read it. And this was in the President. And see, the President, what chapter was that? The President was chapter 10, which means episode 280.a would have included this omitted reference in the Amazon Audible audiobook. In March 1998, more than 50 senior members of the Clinton administration gathered at Blair House to rehearse how they might handle a biological attack by terrorists. It was what the government calls a tabletop exercise. Dick Clark had convened the officials to give them a hands-on sense of the decisions they might face as an infection rocketed through a panicked population. For help in drawing up a scenario, Dick Clark's staff turned to William A. Haseltine, and that's Haseltine, an expert on genetic engineering and the founder of Human Genome Sciences, a company that identified and patented genes for drug development. So a lot of these worst-case scenarios of this biological scare and how it spread person to person and the lethality of it, etc., was being suggested, even though the worst-case scenario didn't necessarily have any scientific merit, by William A. Haseltine, expert on genetic engineering of Human Genome Sciences. So Human Genome Sciences was a biopharmaceutical company founded in 1992 by Craig Venter, Alan Walton, and Wallace Steinberg. It uses the human DNA sequence to develop protein and antibody drugs. It had drugs under development to treat such diseases as hepatitis C, systemic lupus erythematosus, anthrax, and cancer. It talks about the corporate history, and the company was founded by Alan Walton, Wally Steinberg, and Craig Venter, who at the time founded the nonprofit Tiger, that's the Institute of Genetic Research, to begin sequencing and submitting patents on hundreds of thousands of protein-encoded DNA fragments. In 1992, Wally Steinberg hired William A. Haseltine as the first CEO of Human Genome Sciences, where he served as the founding chairman and chief executive officer for the first 12 years of the company. So there is a page here for William A. Haseltine. And sure enough, it's a long page. I mean, it is a really long Wikipedia page. Holy moly. Holy moly. How did I not know more about this person here? William A. Haseltine, he's still alive. He's still active. He studied under David Baltimore. In 2001, he was listed by Time magazine as one of the world's 25 most influential business people. How did I not know about him? But in this case here, we know that he is one of the omitted names in the book Germs. Again, hours before September 11th, 2001, talking about a biological terror attack in a major U.S. metropolitan center. And what it doesn't mention in here, okay, is it just says that William A. Haseltine, he just kind of comes in out of the blue. and it says he's with Human Genome Sciences. It doesn't say that Human Genome Sciences, the book anyways, was founded by Walton Craig Venter, and Craig Venter is also a primary character in the book, and a Wally Steinberg. So right there, right off the bat, there's a little bit of misleading. The book is also hiding, I would have to say it's hiding the relationship between Haseltine and Venter. I mean, it's kind of weird that Venter says you need to be worried about this bioterror attack. That leads to Dick Clark having these get-togethers and these exercises, these tabletop exercises. He brings in an expert, William Haseltine, who is actually the CEO of the company founded by Craig Venter. Craig Venter, who, by the way, curiously, is name-dropped in early 2019 by Stephen Hatfill, a guy who is very, very underestimated. I kind of like Stephen Hatfill. Okay. So, Steinberg, I mean, let's just list it out, okay? 1, Haseltine is censored in the Audible book. 2, Haseltine-Venter relationship is hidden in both the book and the audiobook. Another thing that is censored here is on Wikipedia here. The company was founded by Alan Walton, who there's a Wikipedia page for. Wally Steinberg. No Wikipedia page for him. And Craig Venter. I've seen this done before, okay? As a matter of fact, there's another time I've seen this done on Wikipedia where it referred to a man named Dick McKernan. McKernen. Odd, because that other page that referred to a man named Dick McKernan was actually trying to refer to a Richard McKernan, who was the father of Kevin McKernan. So sometimes Wikipedia, you'll see typos left in there, or people refer to with an informal first name, which almost nobody ever used. Richard McKernan, Kevin McKernan's dad, was never called Dick McKernan. He's called Richard McKernan, or Rich McKernan. Similarly, Wally Steinberg. There is a man named Wallace Steinberg, and for whatever reason, Wallace Steinberg does not have a Wikipedia page. Of course, that's where we come in. So if you go to Houstonic.live, Steinberg, Wallace Herbert Steinberg, there he is, born in 1934. In fact, here he is playing tennis. Here he is playing tennis with Geraldo Rivera. I find that to be funny because Geraldo Rivera actually did an interview with Kevin McKernan on the day that they announced the completion of the Human Genome Project. That's funny. But anyways, Wallace Steinberg and Wallace Steinberg, who was never called Wally Steinberg. Wallace Steinberg died on July 26, 1995. And I'm going to say that his death was sudden and curious. He was actually the chairman of the Healthcare Investment Corporation, the largest venture capital fund devoted to healthcare. And he died in his home, I'm going to say unexpectedly, at age 61. So the money to start the money that was used to start this project called Human Genome Sciences, which, sequenced anthrax and did all of these other things, which were so important to the preparations of a potential large-scale biological terror attack in a major U.S. metropolitan center. Technologies which were used as part of getting ready for DNA vaccinations and so on. Technologies which helped make this William A. Haseltine, censored from the book, so rich. The startup capital came from a man named Wallace Steinberg, who never was called Wally. And Wallace Steinberg got a large New York Times obituary. When you get a New York Times obituary that's half a page, okay, you are then at a level, in half a page in the New York Times. If your obituary is half a page in the New York Times, I think you are at a level where you automatically should have a Wikipedia page. Now, I know no one's ever done a correlation of obituary size to Wikipedia pages, but if the New York Times has enough to say to fill half a page with your obituary, you should have a Wikipedia page. All right? I think most people would agree with that. But this is one of those odd cases where there is no Wikipedia page for Wallace Steinberg. No one wants to talk about him. And he provided the startup money for human genome sciences. Of course, all of this stuff is censored in various stages. Some of it is censored from the book Germs. And then after there was no large-scale biological terror attack, I guess they decided to censor even more in things such as the Amazon Audible audiobook. So Wallace Steinberg is, and William A. Haseltine is censored. Now, I've not really heard anyone ever talk about William Haseltine. Okay? Sometimes, I mean, if a name comes up a lot elsewhere, I will use that as a suggestion that maybe I should dig in. Of course, people talk a lot about some individuals like Jeffrey Epstein, who, I'll tell you what, the less you know about the Jeffrey Epstein, the more progress you make in your research. It's true. Some people are put out there. Their visibility is heightened precisely to keep people busy and to reduce the quantity of seconds and cycles that they have to think about other things. And Jeffrey Epstein is one of those things. You have your, what does Jonathan Cooey say, 10,000 thoughts a day? If they can get you to waste 100 or 1,000 or 10,000 of those, or whatever it is, that would be all of them. I guess that couldn't happen. But as many of them as possible on something that is a path to nowhere, that's where they want citizen researchers to focus. And I opened up Whitney Webb's 2 books, Search for Haseltine, and it wasn't mentioned. So he's not mentioned because they don't want you to think about him. And here he is. He's in the Germs book. Everyone in the Germs book I want to know more about. He's worked for David Baltimore, and he's still kind of in the mix. He's definitely still active. So now I have to add Haseltine to my my list. So I have a Wikipedia page right now for William A. Haseltine. Let's see here. We can go up to the H's. H. The page I have is very messy, so I think we're going to be spending most of our time actually sticking on the Wikipedia page. But I just have a lot of links in there right now on this William A. Haseltine page. You can find a bunch of very interesting presentations for William A. Haseltine on C-SPAN still, okay? You can find him, you see, look at these. So this is a year 2000 video where he's on C-SPAN. He's taking the, calls from the audience here. He's talking about the future of genetics on a board on May 15th, 2000. But I think one of the most interesting videos of him, and I may actually play this one a little bit here, is, in a moment anyways, is a 1997 video titled The Promise of DNA Research. Now, this video was on June 30th, 1997. It should be noted that he's presenting early in the morning, and then not long after him on the same day, another scientist presents named Dr. Margaret Liu, L-I-U, 1997. And Margaret Liu, of course, is one of the more prominent characters in the Big Shot AIDS vaccine book. She's done work with a bunch of Robert Malone associates, including Gary Rhodes and I believe John Wolfe to some extent, and Nancy Hagewood. Nancy Hagewood, that's right, the person who accused Bruce Ivins of being responsible for the anthrax letters. This is all within the network of Margaret Liu. And Margaret Liu, very first thing she does in her presentation, she talks about DNA vaccines, is credits William A. Haseltine with a bunch of things. Margaret Liu, noteworthy that she studied under the big guy at Merck Corporation who's responsible for more childhood vaccines than anyone else, Maurice Hilleman. She studied under Maurice Hilleman. And so William A. Haseltine, he talks here, and I think his first 5 minutes is pretty chilling. And in fact, it lays out exactly, I believe, what only a few people have been capable of summarizing for you. Very few people have been able to communicate how important it is to know how central the pursuit of the knowledge of the human genome is. And it's not just the human genome, it's the pursuit of knowledge related to how to turn life into an algorithm which can be manipulated and modified and augmented. Turning life into something that people can take control of, allow humans to take control of their own evolution. I mean, it's one of the biggest it is the biggest scientific endeavor of all time, the longest running and the most well-funded scientific endeavor. And it's not just the Human Genome Project, it's everything. It's all AIDS, HIV research, vaccine research, going all the way back to World War II and the Manhattan Project. And it continues up until the current with, of course, bioterrorism and COVID-19. So William A. Haseltine presents here, and he seems to also, in other videos you can watch, have a pretty good working relationship with Anthony Fauci. Again, thank goodness, the Germs book tells us this is what we should be thinking about going into the fall of 2001, where everyone's expecting a large-scale biological terror attack in a major U.S. metropolitan center, which, according to the book, is going to create the perfect opportunity to unroll gene therapy, DNA vaccines, to get over the hump that was set by the FDA. And they were going to come up with a justification to use them on the United States military because they couldn't make the old vaccines fast enough. William A. Haseltine has written a variety of papers. One paper that I will highlight just to get things started here is a 2024 paper called RNA Structure, Past, Future, and Gene Therapy Applications. All right, and this is William A. Haseltine, Kim Hazel, and Robert Patarka. I don't know who that is. But William A. Haseltine's the primary author here. And again, this is 2024. So the abstract is first believed to be a simple intermediary between information encoded and DNA and that functionality displayed in proteins, RNA, is now known to have many functions through its abundance and intricate, ubiquitous, diverse, and dynamic structure. About 70 to 90% of the human genome is transcribed into protein coding and non-coding RNAs as main determinants, along with regulatory sequences of cellular populations of cellular to populational biological diversity. Blah 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 blah. So it talks about the history of RNA. And what I find interesting here is while going through the history of RNA, talking about Watson and Crick and Franklin and Gosling, right around, let's see here, right around, I think it's page 11 in here, the story of mRNA therapeutics dates back to the early 1960s when mRNA was discovered as a critical player in genetic information flow. However, it was in the late 1980s that researchers began exploring mRNA as a therapeutic tool. In 1987, Robert Malone from the Salk Institute demonstrated that synthetic mRNA strands mixed with lipid particles could transfect human cells to express proteins of interest. That is a pretty grand statement there. So, in a way, this document by William Haseltine has, I think, several ulterior motives, and I think one of those is to reinforce this mythology that Robert Malone is a genetic mastermind. I believe Robert Malone is a very smart person, but I've worked in the fields of engineering and sciences, and it's very seldom that someone just walks over into the corner and has 10,000 unique thoughts all of their own. Usually things are built upon other people. They're the result of nudging or slight increments, slight innovations upon the ideas of others. So this idea that Malone, hey, I'm going to go do something wacky over here at the Salk Institute is it's mythology. I'm sorry, it's mythology. He was encouraged to do this probably at the behest of Bob Gallo, Murray Gardner, and several other people connected with the history of the Special Virus Cancer Program and so on. Either way, this William A. Haseltine, again, a name censored, seems to have a very odd, every once in a while, bump in with the history of Robert Malone. So I wouldn't be surprised, given the fact that Haseltine is, of course, a very pro-Fauci person, pro-mRNA, etc. You would see someone like William Haseltine never mentioned by, of course, a Whitney Webb, because a Whitney Webb is, I believe, in cahoots or paid for, something like that, some arrangement, by Robert Malone. Robert Malone brought her to the first Children's Health Defense Conference, and it would make sense that Robert Malone would probably guide Whitney Webb to, hey, just don't mention Haseltine. It's not going to go anywhere good, honey. Just don't mention that. I'm not saying Robert Malone would talk like that. Maybe he would. I don't know. So I want to know more about Haseltine. Why is he censored? If you want to, well, first and foremost, we're going to go over the family of Haseltine in a few minutes as well. Haseltine has a sister. She is one of the most vocal people right now and influential people with respect to gender rights and gender fluidity, etc. Of course, gender having an impact on infectious diseases. That's William A. Haseltine's sister. And, oh my goodness, his brother, Eric Haseltine, is on the board of the United States Technology Leadership Corporation. Get a load of this one. He was 13 years at Hughes Aircraft, rose to position of Director of Engineering. He then left for Walt Disney Imagineering in 1992. He joined the Research and Development Group working on large-scale virtual reality projects. In 1998, he was promoted to Senior Vice President responsible for all technology projects. In 2000, he was made Executive Vice President, Virtual Reality, Walt Disney. He was then Head of Research and Development for Walt Disney Imagineering. By the time he left in 2002, he then joined the National Security Agency as Director of Research. He was the Director of Research for the NSA. That's right, William A. Haseltine. In the book, Germs, his brother, Eric Haseltine, becomes the Director of Research for the NSA, the National Security Agency. And then from 2005 to 2007, Haseltine was Associate Director for Science and Technology, Office of the Director of National Intelligence, ODNI, that organization's first, a position he described in 2006. U.S. News and World Report interview by saying, "You can think of me as the CTO of the intelligence community." Okay, so you got the CTO of the intelligence community. You have the person who's the CEO of Human Genome Sciences, referencing Robert Malone as, "Hey, well, that guy definitely did the mRNA stuff." And then you have the sister who's in some serious leadership positions related to, gender fluidity and gender and genomes. And I never knew about this family tree before. It's all over the place. Now it's stuck to me like a wet paper towel. So what I wanted to do is just play a couple of excerpts here from this one interview in particular, the promise of DNA research. Okay.
Institute, and I'm delighted to welcome all of you here this morning for this AEI seminar on the promise of DNA research.
Our first speaker for the day is Dr. William Haseltine. Dr. Haseltine is Chairman of the Board of Directors and Chief Executive Officer of Human Genome Sciences, Incorporated. He holds a doctorate degree from Harvard University in biophysics and has been a professor at the Dana-Farber Cancer Institute of Harvard Medical School and at the Harvard School of Public Health.
Note, she said physicist. And by the way, his dad, William Reed Haseltine, who I can't find too much information on, was also a college physicist. And he also had some role with respect to ballistics in United States Navy research.
He has published over 250 scientific articles is the editor-in-chief of the Journal of AIDS and has founded 7 companies, each in a different area of medicine.
He was the head of the Journal of AIDS. Okay. Again, he was the head of the Journal of AIDS. He is featured prominently in HIV research. And he then transitions into being afraid of biological terrorism. Just like all the people of the Human Genome Project, such as Craig Venter, Claire Fraser, many others, are all suddenly afraid of bioterrorism. You know why? Because biological terror, or a biological terror attack in the major U.S. metropolitan center, if it's big enough, is going to create an opportunity to use DNA vaccines, research which comes out of AIDS, on the United States military. This is why no one out there will read these 2 books. These are the reading and the research of these 2 books. I promise you, right now, will give you more insights into the nature of what happened for September 11th, 2001 than any other 2 books in the world. Period. And you need to deep dive on both of these books. You need to circle every name, every company, and make pages for them. These 2 books. How crazy is it that the HIV research of Nancy Hagwood, not only is she the 3rd most frequent co-author of Robert Malone's postdoc advisor, Murray Gardner, I believe Nancy Hagwood may have also worked with Haseltine, and she definitely worked with Margaret Liu, who has another video on this day. I just reposted it. And then she goes to the FBI in 2007 and says Bruce Ivins did it. Come on. Anyways, the physicist part is important because it was during the Manhattan Project in World War II where Schrodinger and really physicists such as Max Delbruck and Leo Szilard and so on started to try to say, okay, how are we going to, or can we now, describe the human body as an algorithm? Can we just come up with the data? Because it's nothing more than it's physics. We can break the human body down. We can break life down to algorithms. They all transitioned into biophysicists, molecular biology, etc. So a lot of the early molecular biologists started off as physicists, not really biologists, physicists. They were trying to turn life into algorithms. And how was this protected? Well, it was all protected under the cloud of the atom bomb. Really. I mean, I don't know how else to say it. You know, the Atomic Energy Commission and the Department of Energy controls the Human Genome Project. How crazy is that? Is it because, I don't know, they just had a few people available? Or is it really the core mission to provide a level of secrecy to use the general population for the purposes of medical research, data collection, and so on? That's more likely the case. And you can tell people they can't know what's going on because of nukes. Anyways, biophysicist, she says.
Past 16 years. Last year, he received the Ernst & Young 1996 Greater Washington Entrepreneur of the Year Award for Biotechnology. Dr. Haseltine will be presenting an introduction to the basic science of DNA research. Please join me in welcoming him.
It's a great privilege and pleasure to be here today to introduce this very interesting forum for the American Enterprise Institute. The topic is one of great interest, not only to professionals who are interested in the fundamental aspects of biology and medicine, but also to all of us as citizens who are concerned about our health and our environment.
And just to remind you, this is a 1997 video, and here's William A. Haseltine today. This is the current Wikipedia page, almost 30 years later.
Because just as we look at our beautiful world today, with its blue skies and its green rolling hills, its lush vegetation and its rich variety of life under the sea, we have to come to realize that.
And they spelt his name wrong. It's Hasel. It's not Hazel like the color. It's Hasel. Haseltine, not Haseltine.
That all of that was created by living forms from what was essentially a barren rock with water on it. How did that marvelous transformation come about? The very air and oxygen we breathe was not in our atmosphere before there was life on Earth. What happened was living forms evolving over time created first a simple chemistry, then a more complex chemistry that has, in the words of the science fiction writers, terraformed this rock to give us what we now live in as a most pleasant environment. That's how powerful the chemistry of life is.
What did Jonathan Cooey say 2 days ago? Evolution because of DNA. The chemistry of DNA. If you sprinkle a little DNA on a planet, the planet can be terraformed. This is the guy who's advising President Clinton and is being used by Dick Clark to basically scaremonger all of the intelligence agencies and the U.S. military about the capabilities of biological terrorism going into the 21st century. Mentioned in this book that's who they bring in as the expert. You think DNA is scary? You can terraform a planet because of DNA. That's how deep the scaremongering is, and the cult is. It's a very powerful thing. To just make that statement. You don't need a god. All you need is a little DNA. That chemistry is so incredible. Throw it on a planet. Boom, it's terraformed, just like a Star Trek movie. Go back a little bit here. It's an amazing statement.
We have to come to realize that all of that was created by living forms from what was essentially a barren rock with water on it. How did that marvelous transformation come about? The very air and oxygen we breathe was not in our atmosphere before there was life on Earth.
Wow.
What happened was living forms evolving over time created first a simple chemistry, then a more complex chemistry that has, in the words of the science fiction writers, terraformed this rock to give us what we now live in as a most pleasant environment. That's how powerful the chemistry of life is. It is powerful enough to transform an entire planet from a bare rock to what we see today. What modern biology has done is allowed us to peer very deeply into the nature of living things, to peer beyond what is visible to the naked eye, to peer beyond what is visible to the microscope, and today, with our modern understanding of atomic science and our modern understanding of how living organisms are built, we can at last unite what have been the two great trends of science, the theoretical understanding of what nature is and how it comes to be from the first principles ranging from energy, conservation of mass, the nature of the atom, to what a living organism is. It's been my great privilege as a scientist to be part of the revolution in human understanding that leads to that great fusion. I am trained as a biophysicist. Think about that for a minute and what it means.
He stresses
it means a physicist interested in biology,
He stresses
or a biology interested in physics, a biologist interested in physics, or a fusion of the two. And indeed,
Schrodinger, Teilhard de Chardin.
we can now begin to understand the living world in terms of the atoms, the chemistry that make up all of what is in this universe, and therefore make up that which is life. That may seem a far cry from creating better means to prevent.
I've never heard someone say it so succinctly. I've never heard someone say it. Just put it out there so succinctly and completely. To be clear, I'm not saying I agree. I mean, I think this is nuts. But of what has happened, and he's comfortable enough to just say it here in 1997, because that's exactly what has happened. This time, 1997, we're basically 50 years after the atom bomb right now, okay? And he says energy, physics, atoms, taking control of human evolution, being able to terraform planets with this knowledge. This is who the Pentagon is listening to right now, thanks to Dick Clark.
To predict, to cure, and to treat diseases, but in fact it is very closely related. Let me give you a...
Because they're going to use bioterrorism and infectious diseases and the need to use people, to subject people to vaccinations as a route to learn more to accomplish their goal and their vision. It's not that they learn more about the human genome or body or whatever, and they can protect us better. No. They need us to be subjected to the vaccines and to the data collection so they, in turn, can accomplish their goal. Because humans provide the most valuable resource for what they need. They will never be successful without humans. If it was just these genome guys, okay, I don't know, on an island somewhere, no internet, they wouldn't figure it out. They need billions of human beings, of test animals to collect data from, right? I mean, does it make sense? How do we learn more about pharmaceuticals right now? We test them on animals. How can you advance knowledge of the human genome without humans? You can't. Humans are the most valuable resource. And these people have incredible power. But they don't have enough power to buy people. So we need to create a mythology that everyone's going to subject themselves to, from bioterrorism to infectious diseases. So people volunteer the only thing they really have in the world, which is their bodies, for the purposes of them accomplishing their goals. And they don't have to spend any money to do it. Now, if they wanted to be transparent, they wanted to say, look, we think this is really valuable. We're going to, if you want to enroll in this, we're going to give you, $1 million. And if you die unexpectedly, there's going to be $10 million payout to your surviving family or something like that, right? Come up with a price. So people say, okay, I'm willing to go along with that. No, they don't want to do that, because that would mean the value of the U.S. population is thousands of trillions of dollars. They don't have that kind of money, or much less the world. I'm just going to play it again. I'm so happy at how succinctly he says it all.
About our health and our environment. Because just as we look at our beautiful world today, with its blue skies and its green rolling hills, its lush vegetation and its rich variety of life under the sea, we have to come to realize that all of that was created by living forms from what was essentially a barren rock with water on it. How did that marvelous transformation come about? The very air and oxygen we breathe was not in our atmosphere before there was life on Earth. What happened was living forms evolving over time created first a simple chemistry, then a more complex chemistry that has, in the words of the science fiction writers, terraformed this rock to give us what we now live in as a most pleasant environment. That's how powerful the chemistry of life is. It is powerful enough to transform an entire planet from a bare rock to what we see today. What modern biology has done is allowed us to peer very deeply into the nature of living things, to peer beyond what is visible to the naked eye, to peer beyond what is visible to the microscope, and today, with our modern understanding of atomic science and our modern understanding of how living organisms are built, we can at last unite what have been the two great trends of science, the theoretical understanding of what nature is and how it comes to be from the first principles ranging from energy, conservation of mass, the nature of the atom, to what a living organism is. It's been my great privilege as a scientist to be part of the revolution in human understanding that leads to that great fusion. I am trained as a biophysicist. Think about that for a minute and what it means. It means a physicist interested in biology, or a biology interested in physics, a biologist interested in physics, or a fusion of the two. And indeed, we can now begin to understand the living world in terms of the atoms, the chemistry that make up all of what is in this universe, and therefore make up that which is life. That may seem a far cry from creating better means to prevent, to predict, to cure, and to treat diseases, but in fact it is very closely related.
Of course it is.
Let me give you a story of what we are doing at Human Genome Sciences and how that fits into this emerging picture of what we are.
I haven't watched the rest of this yet, by the way.
When I was a student, I worked for James Watson, the man who discovered DNA, and Walter Gilbert, the man who learned how to read the text of DNA.
And that would mean he also, at some point, worked with Sidney Brenner, who doesn't have this type of fanatical assessment of the situation.
At that time, we thought of the living world as relatively simple. There was an information molecule called DNA, which was the storehouse of individual genes. Genes are the irreducible, heritable traits. They were originally defined by Mendel and then by others based on observation, that the earlier concept that inheritance was a matter of mixing of two fluids, and we still have that in the concept of bloodlines. If you say it runs in the blood, or this is characteristic of a pure blood, or that kind of blood, we still have that nomenclature. He actually defined, through very careful observation, the fact that genes didn't behave like fluids. They behaved like two bags of marbles that were actually fundamentally immiscible, but had different traits associated with them. And one of those irreducible heritable traits for height, for color, for whatever it was he was measuring, actually didn't mix with something, but could be separated out in subsequent generations. That's what the origin of the gene was. But later, people came to understand what the physical basis of the gene was. That was a theoretical construct. But during the 50s, and with the discovery of DNA, people came to understand that a gene, this heritable trait, had a physical reality. It was a length of a molecule called DNA, which for all practical purposes you could think of as a two-stranded rope. It's a long molecule with two different strands that has the wonderful property that when you separate the two strands, a new and identical strand can be templated on the other to make two essentially identical ropes. Well, what is that rope doing, and how does it make a gene? That rope is like a piece of magnetic tape. It contains information. It has chemicals built into it, 4 different chemicals, which actually spell out a code that contains information. And that code, when processed by the living cell, produces from one length one product, which is a protein. So at the simplest level, what you have and an understanding of genes is one gene is an immiscible trait that is inherited, and at the other, the molecular atomic level resolution.
Transcription.
One gene is a length of DNA made of atoms which contains information that when processed by the cell makes one protein. One gene, one protein. Proteins do three different things in the body. Proteins build structures, they build the fibers, they build the physical structures of our body. They are the enzymes that transform one chemical to another. The breakfast or lunch that you're now digesting, depends when you listen to this, is now being transformed by enzymes from one chemical form to a different chemical form by those enzymes. And the third function of a gene is a switch. It turns systems on and off. The most familiar switches to most of us are hormones that act throughout our bodies to turn on or off different functions, but we have a lot of different switches. Altogether, we now understand that a human has, and it may seem small or it may seem large depending on your point of view, about 150,000 different pieces, different genes that make up that collection of a human. So that's where we've progressed up to now.
Of course, he doesn't mention the microbiome.
And what happened about 5 years ago was a fusion, as very often happens in science, a fusion of a number of different technologies which allowed us to gain a much more rapid and deeper insight into how we are built at the genetic level. We had this theoretical construct of what a gene is, of how it was made, that a gene is a protein and a protein has these properties, but that was mostly achieved by the study of simpler organisms. When it was applied to humans, it was applied one at a time, and when you're trying to find 150,000 things one at a time, and each one takes between, say, 2 and 10 years to find, you can't find very many of them. As often happens in science, 3 different technologies came together to make the science of gene hunting much quicker, more rapid, and indeed almost automatic. And what happened is over the process of about 25 years, people began to learn how to work with genes. At first it was a rare art form, it was the one I was trained in. Later it became easier, and over time people learn more and more tricks, sort of like cooking tricks, of how to work with genes.
Well, I would recommend watching the rest of it.
A related revolution in robotics. We as humans.
This, of course, is all a big part of making the Human Genome Project possible, because the Human Genome Project is central to the biological terror attack expectations in a major U.S. metropolitan center. And as the book shows, on one page, it just says it outright. DNA vaccines are going to be the thing which allow us potentially to deal with this because of all of the problems with that old anthrax vaccine. It should be observed, for example, if you look at the Whitney Webb literature, she focuses exclusively on the old anthrax vaccine and how it was run by Zionists and Muslims and Brits and all those people are bad people and greedy people and how they stood to make millions of dollars from 9/11. Which is exactly my narrative. They wanted to demonize the old anthrax vaccine as much as possible and skip criticism of the new technologies. Matter of fact, Whitney Webb proves my hypothesis by what she talks about extensively and what she doesn't. Who's the guy who wrote that Emerging Diseases book? Oh, yeah. Horowitz. The Horowitz guy proves me. The AIDS and Ebola guy, he proves my point because he goes on and on and on about how bad the old anthrax vaccine was, never mentions Lederberg, and doesn't even really talk about Vical Corporation. It's all the same. They all do it because they're all paid to lie and not tell you the full truth. Or they're paid and said, only talk about this, don't talk about that, and they don't know they're lying. One or the other, I don't know. I shouldn't be presumptuous in their lying, but, I can be a little bit critical, maybe even more so, because, again, their unwillingness, the majority of independent citizen journalism, to talk about the drug epidemic in the United States of America and put it into perspective makes them complicit in murder. I'm not talking about 5 deaths, 5,000, 50,000, even more than 500,000. We're talking about a statistically significant percentage of the U.S. population. And you don't have... Anyways, I'm not surprised. Long story short, I'm not surprised Haseltine was censored. There's a few videos on him out there. There's a recent interview between him and the previous FDA director, Margaret Hamburg. Margaret Hamburg, of course, also in the Germs book. New York City Health Commissioner, getting New York City ready for a biological terror attack. I don't know how many more connections I could possibly put out there to prove my point. It's just the way it is. It's just the way it is on this channel. All right, guys. Thank you. I need to move along. I hope this was a useful morning for you. We'll do more. We'll do more soon. Love you. Thank you. Take care. God bless.